Bismillahirrahmanirrahim. Assalamu alaikum students. We are going to start our new chapter, multicellular and tissue level of organization. Here we are discussing about the evolutionary perspective. Animals with multicellular and tissue level of organizations have captured the interest of scientists and the laypersons alike. A description of some members of phylum Cnidaria, for example, could fuel a science fiction writer's imagination. एक साइंस फिक्शन राइटर है जो कि इमेजिनेट कर रहा है फाइसेलिया फाइसेलिस के बारे में, यू कैन से निडेरियंस के बारे में. उसकी राइटिंग्स कुछ इस तरह की है. फ्रॉम अ डिस्टेंस आई वाज नेवर थ्रेटन, इन फैक्ट आई वाज infatuated with its beauty. A large, inviting, bright blue float lured me closer. As I swam nearer, I could see that hidden from my previous view was an infrastructure of the tentacles, some of which dangled nearly 9 meters below the water surface. The creatures seemed to consist of many individuals, and I wondered whether or not each individual was the same kind of being, because when I looked closely, I counted 8 different body forms. I was drawn closer, and true nature of this creature was painfully revealed. The beauty of the gas-filled float hid some of the most hideous weaponry imaginable. When I brushed against those silky tentacles, I experienced the most excruciating pain. Had it been for my life vest, I would have drowned. Indeed, for some time I wish that had been my fate. इसमें जो है एक साइंस फिक्शन राइटर जो है वो मेंशन करना है फाइसेलिया फाइसेलिस के बारे में, उसकी ब्यूटी के बारे में, कि जब वो पानी के अंदर था तो उसको एक लंबी फ्लोटिंग चीज़ ने जो है उसको अपनी तरफ अट्रैक्ट किया. जब वो करीब गया तो उसने देखा कि 9 मीटर्स जो है वो लंबे टेंटिकल्स वाली कोई चीज़ है जो कि सरफेस से नीचे तक 9 मीटर्स की लंबाई में आते हैं. फिर जो है उसने कहा कि ये देखा कि वो जो क्रिएचर थी वो मैनी इंडिविजुअल्स पे कंसिस्ट कर रही थी, और जो है जब उसके बहुत करीब गया, जब उसको टच किया, सिल्की टच आया टेंटिकल्स का, तो पेन स्टार्ट हो गया. उसे पेन फील हुआ, लेकिन वो पेन उसको उसकी ब्यूटी के साथ बहुत बेहतर लगा. यानी जितनी उसकी ब्यूटी है तो वो उसको पेन जो है इतना फील नहीं हुआ ऑन सीइंग द ब्यूटी ऑफ द फाइसेलिया फाइसेलिस और द मेंबर्स ऑफ phylum Cnidarians. Swimmers of tropical water who had come into contact with the Physalia physalis, the Portuguese man of war, know that this fictitious account rings true. कि जो उसने fictitious लिखा है, fictitious जो साइंसी राइटर्स होते हैं या कोई राइटर होते हैं वो अपने जहनी ख़यालात की वजह से लिखते हैं, जो ख़यालात उनके आइडियाज़ बन जाते हैं. जो उसने लिखा है that is true. In organisms such as Physalia physalis, the cells are grouped, specialized for various functions and interdependent. These phyla are the Porifera, Cnidarians and the Ctenophorans, in which we discuss about the multicellular organization that varies from loose association of the cells to the cells that are organized into 2 distinct tissue layers. Portuguese man of war कहा जाता है पुर्तगाल. Here is the diagram 9.1. That is the Physalia physalis, the Portuguese man of war. The tentacles shown here can be up to 9 meters long and are laden with nematocysts that are lethal to small vertebrates and dangerous to humans. A bluish float at the surface of the water is about 12 centimeters long. It is not shown in this photograph. The entire organism is actually a colony of polypoid or medusoid individuals. Now next topic is the origin of multicellularity. Multicellular life you can distinguish between a unicellular cell and that of the multicellular cell. Unicellular consisting of the single cell, while that of the multicellular consisting of many cells. So there are दो opinions हैं कि either जो है multicellularity जो है वो कैसे originate हुई, यानी यूनिसेलुलर से एक सेल से काफ़ी सारे सेल्स का जो ओरिजिन है वो किस तरह हुआ, ये हम जो है हाइपोथेसिस जो है वो डिस्कस करते हैं। Multicellular life has been a part of Earth history for approximately 550 million years ago. Although this seems a very long time, it represents only 10% of the Earth's geological history. Multicellular life arose quickly in the 100 million years prior to the Precambrian or the Cambrian boundary, in which scientists view an evolutionary explosion. कैम्ब्रियन एक्सप्लोजन कहा जाता है, जिसके अंदर एकदम से डाइवर्स ग्रुप ऑफ ऑर्गेनिजम्स जो हैं वो इवॉल्व हो गए. These evolutionary events resulted not only in the appearance of all of the animal phyla recognized today, but also 15 to 20 animal groups that are now extinct. कैम्ब्रियन एक्सप्लोजन पहले भी एक्सप्लेन हो चुका है कि एकदम से ऑर्गेनिज्म जो हैं डाइवर्स जो है, यू कैन से कि डाइवर्सिटी ऑफ ऑर्गेनिज्म जो है वो अपीयर हो गई ड्यूरिंग द कैम्ब्रियन पीरियड, इसलिए इसको कैम्ब्रियन एक्सप्लोजन कहा जाता है. ओरिजिनेट हो गए बहुत सारे फाइला, 15 to 20 एनिमल फाइला जो है वो अब उसमें से एक्सटिंक्ट भी हो चुके हैं. सिंस दिस इनिशियल एवोल्यूशनरी एक्सप्लोजन, मोस्ट ऑफ द हिस्ट्री ऑफ मल्टीसेलुलर लाइफ हैज़ बीन वन ऑफ द एक्सटिंक्शन. बहुत जो यानी यू कैन से कि जो हमें रिकॉर्ड मिलना चाहिए वो भी एक्सटिंक्ट हो चुका है. The evolutionary events leading to multicellularity are shrouded in mystery. Most zoologists believe that multicellularity could have arisen as a dividing cell remained together in a fashion of many colonial protists. Although variation of this hypothesis exist, there are all treated here as a colonial hypothesis. You can say सेल अलग अकेले अकेले रहते थे, फिर उन्होंने जो है इकट्ठे रहना शुरू कर दिए. कॉलोनी की फार्म में जब रहेंगे तो वो कुछ अरसे बाद वो मल्टीसेलर की अपीयरेंस देंगे. तो इस हाइपोथेसिस को कॉलोनियल हाइपोथेसिस कहा जाता है. सेकेंड प्रपोज्ड मैकेनिज्म इज कॉल्ड द सिंसिशियल हाइपोथेसिस. द सिंसिशियम इज़ ए लार्ज मल्टीन्यूक्लिएट सेल, बड़ा सेल जिसके अंदर काफ़ी सारी न्यूक्लियाई मौजूद हैं, मल्टीन्यूक्लिएट सेल कहलाएगा. The formation of plasma membrane in the cytoplasm of syncytial protist could have produced a small multicellular organism. अब जो है जब बहुत सारे न्यूक्लियाई मौजूद थे तो हर न्यूक्लियस के इर्द गिर्द साइटोप्लाज्म गैदर हो गया और प्लाज्मा मेम्ब्रेन बन गए, जिसकी वजह से एक सिंगल सेल जो है वो चेंज हो गया सिंसिशियल यू कैन से मल्टीसेलर ऑर्गेनिज्म के अंदर. Both the colonial and syncytial hypotheses are supported by the colonial and syncytial organization that occurs in some protist phyla. यानी कुछ के अंदर प्रोटिस्ट फाइला के अंदर हमें colonial hypothesis की सपोर्ट मिलती है और कुछ के अंदर जो है syncytial hypothesis जो है वो करेक्ट दिखाई देता है. नेक्स्ट इज़ द डायग्राम देखें, द डायग्राम 9.3. टू हाइपोथेसिस regarding the origin of multicellularity. फर्स्ट ऑफ ऑल एंसेस्ट्रल प्रोटिस्ट देखें ए के अंदर, इसके अंदर सेक्सुअल रिप्रोडक्शन हुई, दो फिर कॉलोनियल प्रोटिस्ट कॉलोनी की फॉर्म में आ गया, देन 2 लेयर रेडियल एंसेस्टर बना. इससे जो है बायलेटरल एंसेस्टर और रेडियल एनिमल्स जो हैं, एनिमल्स शोइंग द बायलेटरल सिमेट्री एंड एनिमल्स शोइंग द रेडियल सिमेट्री, जो दो यू कैन से ग्रुप्स जो है इन्वॉल्व हुए, बायलेट्रिया एंड दैट ऑफ द रेडिएटा. This one was the colonial hypothesis. Multicellularity may have arisen when cells that are dividing protists produced remain together. Cell invagination could have formed a second cell layer. This hypothesis is supported by colonial organization of some Sarcomastigophora. The colonial protist and the 2-layered radial ancestor are shown in the sectional view. सेकेंड वन इज़ द सिंसिशियल हाइपोथेसिस. लार्ज सिंसिशियल प्रोटिस्ट है जिसके पास काफ़ी सारे न्यूक्लियाई मौजूद हैं. अब सेल बाउंड्रीज बन गई, हर न्यूक्लियस के इर्द गिर्द साइटोप्लाज्म बन गया. देन बायलेटरल एंसेस्टर जो है इवॉल्व हुआ, बायलेटरल से बायलेटरल एनिमल्स बने और साथ ही जो है वो रेडियल एनिमल जो है इवॉल्व हो गए. सो बी जो है द सिंसिशियल हाइपोथेसिस, multicellularity could have arisen when plasma membrane formed within the cytoplasm of a large multinucleate protist. Multinucleate bilateral ciliates support this hypothesis. पहला जो hypothesis है that is supported by Sarcomystigophorans, while the second hypothesis is supported by the bilateral ciliates. Next is the animal origin. The next topic, the fundamental question arising animal origin is whether animals are monophyletic, derived from a single ancestor, or polyphyletic, derived from diphyletic, derived from the two ancestors, or polyphyletic, derived from many ancestors. You can say there are three hypotheses about the animal origin. Either they are monophyletic, either the all animals are originated from a single ancestor, then we can use a term that is called the monophyletic. All the animal phyla are derived from a single ancestor, the monophyletic view. Diphyletic when two ancestors are present, and polyphyletic when every organisms have a separate ancestor, every group have a separate ancestor, then we can say this is a polyphyletic view. So the problems with the monophyletic view is that all the animal phyla in the fossil record in the fossils appeared simultaneously in the Precambrian and the Cambrian boundary. Therefore, it is difficult to explain that animals are monophyletic. अगर हम कहें कि एक ही ऑर्गेनिज्म से इतने सारे जो फाइला हैं वो इवॉल्व हो गए हैं, तो एक ही जो एंसेस्टर है उससे इवॉल्व होना कैम्ब्रियन बाउंड्री के अंदर बहुत मुश्किल है, इम्पॉसिबल है. And proof of the theory, absence of similarities in animal cell organization supports monophyletic view. Ester formation in most animals, esters are formed during mitosis, you well know. Certain cell junctions are similar in all animal cell. कुछ ऐसी चीज़ें हैं जो ये सपोर्ट करती हैं कि all animals are originated from a single ancestor, or they are the monophyletic. First one is the presence of the esters that are formed during mitosis in most of the animals, and cell junctions are also similar in animal cell, and proteins that are involved in the movement are similar in most animal cell. So most animals produce flagellated sperm. Here are some resemblances that show that are the proof that the animals are monophyletic and they have a monophyletic origin. So this all factors, this all resemblances supports the monophyletic theory. Conclusion क्या हो सकता है? Scientists do not accept this view because it is difficult to explain the origin of these common features. Next is the diphyletic view, when we say that the animals are derived from the two ancestors, and polyphyletic when the animals are derived from many ancestors. Most of the zoologists accept this view. And the next topic is phylum Porifera and the animals that contain spores. पोरिफेरा कौन से एनिमल्स हैं जिनकी बॉडी जो है वो पोरस होती है, सुराखदार होती है. देन वी कैन से दे आर द पोरिफेरंस. द पोरिफेरा पोर बेयर. पोरस का मतलब पोर होता है, फिरा का मतलब टू बियर. द स्पंजेस आर प्राइमरली मैरीन एनिमल्स. इसके अंदर जो मेजर ग्रुप्स हैं, या यू कैन से जो मेजर एग्जांपल्स ऑफ पोरिफेरंस आर द sponges that are primarily marine animals consisting of loosely organized cells. The approximately 9,000 species of sponges vary in size from less than a centimeter to a mass that would more than fill your arms. यानी कम से कम जो है वो 1 सेंटीमीटर से भी कम है, और ज़्यादा से ज़्यादा ये कि हमारे दोनों आर्म्स के दरमियानी वाला जो गैप है उसको फिल कर देता है. कैरेक्टरिस्टिक्स ऑफ फाइलम पोरिफेरंस एसिमेट्रिकल और रेडियल सिमेट्रिकल. या तो कोई उनके अंदर नो अरेंजमेंट, कोई सिमेट्री नज़र नहीं आती, हीप ऑफ द सेल्स, लूजली ऑर्गेनाइज सेल्स, या जो है वो रेडियल सिमेट्री नज़र आती है. द मेजर कैरेक्टरिस्टिक्स 3 टाइप ऑफ सेल्स, पिनेकोसाइट्स, mesenchyme cells, and the choanocytes, or you can say the choanocytes. Central cavity or a series of branching chambers through which water circulates during filter feeding. No tissues or the organs are present in case of the phylum Poriferans. cell types, body wall, and the skeleton, the next topic of the phylum Porifera. In spite of these relative simplicity, sponges are more than colonies of independent cells. As in all animals, sponge cells are specialized for particular function. This organization is often referred as a division of labor. सेल्स जो हैं वो कॉलोनी की फॉर्म में हमें नज़र आते हैं, तो उनके अंदर डिवीजन ऑफ लेबर होती है कि काम की तकसीम जो है वो मौजूद होती है. नेक्स्ट इज़ द सेल टाइप्स. There are three types of cells are present in case of the phylum Poriferans. Pinacocytes, second one is the porocytes, third one is the choanocytes, and the amoebocytes in the mesohyl. First, let's discuss first one. Pinacocytes, these are the cells which make outer layer of the body of the sponge. स्पॉन्ज की जो बॉडी की आउटर लेयर बनाएगी उन सेल्स को हम पिनेकोसाइट्स कहेंगे, एंड द लेयर दैट कंटेन द पिनेकोसाइट इज कॉल्ड द पिनेकोडर्म. दीज आर लार्ज, thin and flat cells. These are present on the outer surface of the sponge, and these cells are contractile. Contract कर लेते हैं. The shape of the some sponges change due to this contraction. Each pinacocyte has thin margin with central bulge. थिन मार्जिन्स होते हैं और सेंटर की तरफ से आउट ग्रो सी बाहर निकली होती है जिसके अंदर न्यूक्लियस मौजूद होता है. सेकेंड वन इज द पोरोसाइट्स. इन द नंबर ऑफ स्पॉन्जेस, some pinacocytes are in a tube-like contractile porocytes. These are tubular cells. These cells regulate water circulation. Opening through the porocytes are pathways for water moving through the body wall. Just below the pinacocyte layer of the sponge is a jelly-like layer that is called the mesohyl. Amoeboid cells called the mesenchyme cells move about in the mesohyl and are specialized for reproduction, secreting skeletal elements, transporting and storing food, and forming contractile ring around the opening in the sponge wall. पहली लेयर है स्पॉन्ज की बॉडी को देखें, जो उसकी आउटर लेयर है that is composed of specific type of cells that is called the pinacocytes, and the outer layer that contains the pinacocytes is called the pinacoderm. Sometimes the pinacocytes are specialized into tube-like structures, a tube-like contractile structure that is called the porocytes that can regulate the water. And just below the pinacocyte layer is a jelly-like layer that is called the mesohyl. Mesohyl contains specific type of cells that is called the mesenchyme cells, and these mesenchyme cells are specialized for reproduction, secreting skeletal element, transporting and storing food, and forming contractile ring around the opening in the sponge wall. And next one is the first layer. We have studied the pinacocytes layer. Second one we have also studied the porocytes. Next is the mesohyl, and the third one is the below the mesohyl and the lining. The inner chambers are the choanocytes or the choanocyte or the collar cell. Choanocytes, funnel side cell are flagellated cells that have a collar-like ring of the microvilli surrounding a flagellum. Microfilaments connect the microvilli, forming a net-like mesh within the collar. On the inner side of the sponge body, a layer is present that is called the choanoderm. Choanoderm contains a specific type of cell that is called the choanocytes. These are the flagellated cells that have a collar-like ring of the microvilli surrounding a flagellum. And microfilaments connect the microvilli, forming a net-like mesh within the collar. The flagellum creates water currents through the sponge, and collar filters the microscopic food particles from the water. The presence of the choanocytes in the sponge suggests an evolutionary link between the sponges and a group of protists that is called the choanoflagellates. Sponges are now the skeleton of the sponges. Sponges are supported by a skeleton that may be consist of microscopic needle-like spikes that is called the spicules. These spicules are found by amoeboid cells that are made up of calcium carbonate, silica, or may take on a variety of a shape. Alternately, the skeleton may be made up of spongin, that is a fibrous protein made up of collagen, that is dried, beaten and washed until cells are removed to produce a commercial sponge. And the nature of the skeleton is an important characteristics in the sponge taxonomy. skeleton की नेचर के ऊपर देखते हुए हम पहचान सकते हैं, हम उनको यू कैन से ग्रुपिंग कर सकते हैं. In case of the sponges we have discussed, it have about 9,000 species. They have asymmetrical or radiosymmetrically body. 3 type of cells are present. Central cavity is present, and no tissue or the organs. 3 type of cells. Histology, it is also called the histology of the sponge. First of all, the first cell, pinacocytes, the layer that contains the pinacocytes is called the pinacoderm. It is present on the outer side of the sponge body, and the pinacocytes sometimes form tube-like structure that are called the porocytes that are helpful in the regulation of the water. And below the pinacoderm is a layer that is called the mesohyl that contain the amoeboid cells or the mesenchyme cells that are important in the formation of skeletal elements, and also specialized for reproduction, transporting and storing food, and forming contractile ring around the opening in the sponge wall. Below the mesohyl is a present choanoderm that contains specific cells that is called the flagellated cells that are called the choanocytes, and that have a collar-like ring of the microvilli surrounding a flagellum, and they are helpful in the feeding processes. And skeleton is composed of many needle-like spikes that are called the spicules. They are either formed of they are either calcium carbonate, silica, or a variety of shapes, and sometimes they are also may be formed of spongin, that is a fibrous protein. And the nature of skeleton is an important characteristic in the sponge taxonomy. Here is the diagram of pinacocytes. Porocyte is also shown. A central bulge is shown, outgrowth is shown where the nucleus is present. And next is the diagram showing the body wall of a sponge. You can see, students, on the outer side a layer is present that is called the pinacoderm, containing the pinacocytes. It also contains the porocytes that water for the water flow, and below the pinacocytes or the pinacoderm is a mesohyl that contain a specific cells, and that are called the archaeocytes, and it also contains the spicules. And next is on the inner side a layer is present that is called the collar cells, or you can see the choanocytes, and the layer containing the choanocyte is called the choanoderm, and it contains a single flagellum also. This is the small part of the sponge body surface. On the next slide diagram is the diagram of a collar cell, or you can say the flagellated cell, that contains the flagellum, flagellar fibrils, flagellum, and the mesh-like structure, the collar, and on the inner side nucleus is present. And next is the microvilli is shown, choanocytes flagellum, choanocytes collar, and choanocytes body, and choanocyte nucleus, amoeboid cell in the mesohyl, and trapped food particles in the microvilli, and food vacuole is formed, and amoeboid cell in the mesohyl. This is the diagram showing the structure of a body wall of a sponge, and structure of a collar cell or the flagellated cells. You can better understand these diagrams now.